I'm from the University of Cambridge, and what I want to talk about is some of the work that we've been doing in adapting plant systems for engineering and some of the technology that we've been assembled to deal with this. Now, we think of plants and we think of these quite sessile organisms sitting in place, and one often comes back with the impression that these are quite, well, not very exciting organisms. In day-to-day business, we don't really see the kind of dynamics that you see during morphogenesis. And in this next slide, I show an image, a time-lapse image from the BBC Natural History Unit. And what we're looking at here is a stolon, an extension of an existing leaf, which is growing as an extension here, and you'll see as the movie continues, you see this small organ at the end of the leaf expanding and developing into a closed vessel. So this is now a hollow organ which is continuing to expand and forms a very precise morphology. You can see this expanding now. And it will turn into the mature pitcher of a pitcher plant, where this organ is quite precisely arranged and organized to form this pitcher for this carnivorous plant, and this top portion of the pitcher will just pop open, and you have, after a day or two of development, this precisely engineered structure. And I use this as a way of illustrating this capacity of plants, and of course other organisms, to produce these defined and precise architectures. And they make these kind of structures by virtue of individual cellular processes. So here we have a cartoon of a prototypical plant cell undergoing division. So that plant, after elongation, will divide at an appropriate time, and the new wall that's formed within the plant will be built internally within the body of the existing maternal cell. And so this new wall essentially subdivides the cell to produce the two daughter cells. And these features of plant expansion and division are quite unique to plants compared to animals. So for example, these cells, just like at the macroscopic level, as we see the plants don't move, plant cells themselves within the body of the plant are immobilized. And we have this extracellular matrix which joins and links individual cells within a tissue and means that they can't move with respect to each other. And so unlike animal systems, in plants you have this immobilization of cells. There's no cell migration or apoptosis for that matter. So cells are essentially trapped in place next to their neighbors, and they must retain very plastic fates during development. And despite this, they can produce very precise architectures. So just as we saw the pitcher plant develop in time-lapse motion, we see here that the end products of differentiation, where we have terminal structures, floral organs, that have been developed in very precise ways from these different terminal organs, all produced by these processes of cell proliferation, elongation, and differentiation. Now, one can paraphrase many years' work in molecular genetics in plants to come to some kind of overview of how these things undergo this kind of morphogenetic process. What happens to these cells? What controls cells during this process? And it's paraphrased in this cartoon here, where we have individual cells here which might be programmed to divide at a certain point in development, where we have two daughter cells which are the result of this division process. Now we know from molecular genetic studies in plants that individual cells are propelled in a particular trajectory or particular differentiation pattern by virtue of positional information that's coming from their neighbors. So individual cells are dependent on their neighbors for their individual fates. And as a cell divides and produces two daughter cells, those two daughter cells are by definition in different environments. So they can then pick up different positional cues, have different fates as indicated by these different colors, and of course contribute different information back to those same neighbors. So you have this feedback and very highly parallel system where we have not just one cell doing this, but literally millions of cells doing this in a coordinated fashion during the development of something like a pitcher plant. So in order to get to some better understanding or better tools to engineer this system, we've taken a couple of approaches. And in this talk, I want to talk about first design and analysis and then construction. We think of those two broad issues of attempting to engineer a system like this. And in terms of design and analysis, there's really two issues to deal with. One is actually visualizing the cellular architecture and cell interactions, and the other is being able to link those features together in some kind of computational way to capture the dynamics of those kind of feedback-regulated interactions. So what I want to talk about broadly are these issues of visualization, both of architecture and gene expression. So if we're thinking that these things are underpinning morphogenesis, how do we visualize them? So in the late 1800s, most of the current optical methods that we use were at least established, and here we show a slide from a collection that we have in our department, maybe 100 years old, under what's a modern confocal microscope. So using modern optical instruments, one can get much better images of both algal systems. And this shows fucus here. It's an brown algal system where individual cells are marked out here. This is the cross-section through the top portion of a plant. This is an angiosperm. And we have here the very regular symmetry of the organs which are being produced from the tip of this apex of this plant. And using confocal microscopy, of course, the imaging is trivial to scale and to use high magnification to resolve individual cells inside these thin physical sections of tissues. So we've taken tissues and sliced them physically to produce these kind of images. But of course, a confocal microscope allows you to optically section through intact tissues. So one can take an intact tissue rather than the conventional method of using a microtome. You can use optical sections to give yourself a three-dimensional view of intact tissues. So here we have one of the modern-day model plants, a Arabidopsis in embryo form, which is a very young embryo of Arabidopsis, so-called globular embryo. And each one of these outlined portions here is a cell. So we have it's about the 16-cell stage sitting on top of this stalk here. And around it, we see the maternal tissue. This is still inside the maternal tissue as it's developing. And we're using the microscope to optically section through that tissue to visualize what's going on inside. So using modified techniques that were really established 100 years ago, one can use modified microtechnique to stain tissue, to fix it and stain it in a specific way to label, in this case, the external portion of cells, the cellulose extracellular matrix, and use this in a very precise way to stain those tissues and to use 3D optical sectioning to build up a three-dimensional picture of the arrangements of the fluid inside these tissues. So what we're doing here is to portray the tissue in cell outline form, and of course you can use three-dimensional visualization techniques to give yourself a view of individual three-dimensional shapes within that three-dimensional object. And so using these segmentation techniques, you can seed and inflate or grow that seed to fill a three-dimensional space corresponding to a cell inside that three-dimensional voxel-based, or volume, I should say. So this data set is quite a large data set, might be many megabytes in size, and of course it has essentially doesn't have any value attached to it in terms of labeling or identity of individual features. It has features of intensity, but using these segmentation techniques, you can generate geometric objects which correspond to biologically relevant arrangements, in this case cells, inside the tissue. So here we have that same data set now, labeled and decorated, so we have a three-dimensional view of cell arrangements inside this very early embryonic leaf of Arabidopsis. And of course, you can then build up a geometric view of plant tissues in this way. So we have the embryonic root of Arabidopsis. And of course, these are geometric objects. And so they're numerical and compact. And we can do virtual dissection, for example, to chop away things within the body of the root, to visualize features and intercellular contacts within the root in this case. And the reason why this is so important to us is that positional information, the information that tells cells what they are, is derived by this kind of interaction between cells. And we can use this to gain a qualitative and a quantitative view of the connectivity of cells inside these complicated tissues. Now that's fixed tissue. Of course, in real life, things are living, and this shows a living Arabidopsis root under the microscope. It's highly refractile. Cell walls in plants have a high refractive index. And if you let it grow under the microscope, you see here a time-lapse image. So this root, we've got about 6 hours compacted into this time frame. So at the tip of the root, you've got cells that are being patterned and dividing. And there's quite a sharp transition when they stop dividing and start elongating, and they start differentiating. You see root hairs differentiating on the surface of the root. And underlying this whole process are a whole series of events, including cytoplasmic streaming and exchange of information that takes place. And of course, those are the kind of things that one needs to manipulate if you want to engineer these kind of systems. So the kind of things that we've been doing, and this is work of Fernand Federici, who's in the audience, I think, here we have a Arabidopsis root, very similar to what we saw before, but now decorated with fluorescent proteins. Here we have a red fluorescent protein decorating the nuclei of each cell, and a green fluorescent protein decorating the plasma membrane. And so by a suitable selection of fluorescent proteins and markers that they can be fused to, you can decorate individual portions of the root and now look at, for example, cells as they divide and look at the dynamics of subcellular events within the growing root using confocal microscopy to produce optical sections. So you can see either individual planes or using three-dimensional reconstruction, get a four-dimensional view of the processes that take place within this tissue. Now, I'll just amplify that a little more because what Fernand has done is to make a collection of different color markers, and these markers are being carefully selected to allow counting of cells, which have this nuclear marker, and to allow clean labeling of the boundaries of individual cells. Because as you'll see as we go through, the things that interest us are the way one can alter the negotiation of fates by interaction between cells. So in this way, you can have, say, green and red and blue, red and yellow combinations of markers, which gives you a different view of the individual cell arrangements. And you can use this for two main things. First of all, if you have a mixture of markers, so here we have two different promoters expressing either a green or a red fluorescent protein, mixed and co-expressed inside this Arabidopsis root. We have an optical section here. And you see as you slice through the different cells, you can see both green and red where the two channels have been superimposed on each other. Now, because we're using markers that are both sent to the same place, and these are histone fusions, so they end up in the chromatin, you can then trace essentially a line across the tissue here and quantify the relative contributions of red and green fluorescent protein to individual nuclei. So you can map out patterns of gene expression within the tissue. So this is a one way of trying to deal with some of the issues of promoter standardization, for example, and normalization of expression using ratiometric imaging inside tissues. So that's one thing you can use this for. The second is that you can use these kind of markers for segmentation of live tissue. Now, before we were talking about the production of geometric descriptions of plants in fixed tissue. What we've got here now is live tissue, which can be imaged and then segmented to produce a numerical description of cell arrangements. So using the nuclear markers, one can use this to count individual cells and to provide a seed for segmentation, and have the second marker to mark boundaries of those individual cells. So using these kind of processes, you can mark a seed using the red fluorescent channel, then use the green fluorescent channel as a boundary for a balloon segmentation algorithm, which allows expansion of this domain to map the shape of individual cells to give you a geometric map and a boundary for each cell here. And so that's, for us, is quite a technical step forward. So you can go from images like this, which again is a living root marked with a fluorescent protein. You can mark the centers of cells and then grow the arrangements so we can end up with a collection of descriptions where individual cells are, in an automated fashion, localized, numbered, and have an identity. And of course, quantitative data from those can be gained. For example, in other fluorescent channels, you can get fluorescent image data, which will give you a label or a value for each one of these labeled cells. And so this is important to us because for the kind of things that we want to do, where we want to look at dynamics of interaction between cells, we need numerical models. We need some kind of way of modeling the interactions between these cells. And so what we have used some of this imaging data is to feed into a system, a computational system, which provides a model for morphogenesis and growth in plants. And it's a cellular automata model, so it's based on the description of individual agents or cells which have a genetic script, or that is something which determines cell state and state parameters here. And it does this by virtue of interaction between cells. So these individual cells can produce arbitrary morphogens, and they have individual state parameters which control things like growth, division, turgor, etc. And that these cells interact with each other. But they do that in the context of an extracellular matrix. So this cellular automata, if you like, similar to what you would see in Conway's life, for example, is set within a finite element model. So each of the walls here, the connections between cells, is described in the current version as a viscous object, which is essentially an ortholog of what we see in nature, that cells are separated by this composite structure, the cellulosic structure, which corresponds to the cell wall. And so to try and make that a little more explicit, we have both a biophysical model, which is a finite element model, which underpins this cellular automata, and we have individual cells, each of which has a genetic program running inside it, with interaction between the cells genetically. So you have this time constant, slower time constant of interaction between the cells, and this fast time constant interaction between the biophysical aspects of growth. So cells in one part of the tissue are expanding by virtue of increased turgor and cell wall softening, then that naturally is transmitted very quickly across the model. So you have this biophysical interaction and genetic interaction. And the reason why this is important to us is that if you think of an individual cell, for example, which is growing, in this case it's got a very simple genetic program, so all of these cells have the same program. They're programmed to expand in one dimension, and that corresponds to this red axis here. So they expand in one dimension until they get to about twice their cross-sectional area, twice their volume equivalent. When they get to that point, they undergo cell division. And that corresponds to the blue axis here. And so when they do that, they divide and they go through the cycle again, and they switch 90 degrees between each division cycle. So they make essentially a copy in terms of shape of the original cell, because you've got this alternating division pattern. But as that proceeds, you've got this very simple genetic program interacting with the biophysics of growth. So you have these zigzag arrangements here, which are due to the minimization of forces between the cells. So the biophysical model is constraining the genetic model, and you have an interaction between the two, which gives rise to this characteristic sort of almost bubble-like arrangement of cells inside the tissue. Now this, of course, is quite similar to what you see in real plant tissues, and this kind of minimization of and the arrangement of cells, like formation of 120-degree angles between adjacent cell segments. Now, if you take this kind of arrangement and we have now a morphogen introduced, so these blue cells have that same program. So they're programmed to divide, change axis, divide, change axis. So left alone, these cells would make that sort of rectangular block of cells. But they're attached to cells, so one of the daughter cells here doesn't see that morphogen. So it continues to divide with a different program. So it divides simply in the same orientation. So these cells make a stalk, a column of cells. These cells continue and would attempt to try and make a square, rectangular version. But at the junction of those, because of the interaction between the physical program and these two genetic programs, you end up with this particular morphological feature, which is not programmed explicitly by the genetic program, but rather as a consequence of the interplay between the two. Because cells down here, of course, are constrained physically. They can't expand as they might be programmed to do. And this, I think, is an analog of what is important for us in terms of how one would set about programming cells genetically to make particular morphologies. And the point I'm trying to make here is that you require this linkage between the reality of growth, the biophysical constraints on growth, with the genetic program to do something sensible. So if we have these visualization tools, we need to essentially complement those with ways of manipulating the sequences and manipulating the systems. Now, if we think about plants, particularly crop plants, you think of a diverse collection of things that we would consume or use for human use, and we've got things ranging from from Asia, Middle East, and the New World. We've got corn and beans and almonds, peas, wheat, barley, and bananas, sugarcane, etc. A whole range of quite diverse crops that we use routinely for human uses. And you have this view of diversity, but in fact, we use a very small section of the total plant biodiversity on the planet. A very conservative estimate is that there's something like 250,000 species, plant species on the planet, and higher estimates go up to 400,000. Of those, there are about 20,000 which are known to be edible from human culture, and about 2,000 of these are economically important. About 30 of them produce most of the world's food, and only 3 of them produce more than half of our food. So the grains, the wheat, maize, and rice, produce almost 60% of our calories. So we use a vanishingly small proportion of the total biodiversity. And if you think of existing crops, like corn, for example, here. Corn is derived by almost 10,000 years of human breeding and selection from essentially a weed, Hyacinthe. In fact, most of the crops that exist today have their roots in some weedy crop that early hunter-gatherers domesticated and then shifted over to some kind of agricultural society. And that the transformation of these weeds essentially into modern crops by this gradual process of selection and improvement has produced things which are not biochemically largely different from what they started out from. There's some differences, but really the major difference in terms of yield comes from the agronomic properties and the proportion of cells which are produced in these modern day plants. That it's not so much the underlying biochemical aspects, the subcellular aspects, but it's the way cells are portioned and arranged during development. And so the question is, can you use this as an abstraction, essentially, just as one would in other cases in synthetic biology? Can you manipulate the arrangements of cells rather than the individual constituents within cells? Now, in terms of synthetic biology, we as most in the room would have experience now with standardized components for construction of genetic circuits. And you can think of, in plants at least, two broad classes of application. You can think of taking those kind of systems that one can build and using them to alter cell autonomous circuits, things which are set up and change properties of individual cells, or those which might have self-organizing properties and work exogenously outside the cell or between cells. And it's this latter class of circuits that I want to talk about. And here you can think about things, both symbiotic relationships or things which might change the behavior of collections of cells. So the kinds of things that, certainly in our lab, we're thinking of are things where you could change the arrangements of cells within tissues, presumably similar to what might have happened in human selection of crops, where you've got groups of cells which can be initiated, undergo patterning and differentiation to form different structures. And to take quite a large amount of work and compact it, we've got several systems now for initiation of patterns, which involve foreign transcription activators derived from yeast, which can be hooked into the underlying genetic programs so within a host, in this case Arabidopsis, and can trigger gene expression inside these tissues. And we have several thousand of these lines, which can be used to trigger gene expression in different times and places within inside the organism. And so you can combine these and get different patterns of gene expression expressed within, in this case, an Arabidopsis root. So if you take cell types like this, for example, in the outer layer of the root, you can now program these by misexpressing genes such as this one, which targets proliferation of cells in this outer layer of cells. So here's a wild type root, and here's one where this outer layer of cells has been programmed to hyperproliferate. And certainly you get extra cells within this outer layer, but of course there are also, these cells are connected to each other. You change one type of cell, and there are knock-on effects. So you end up with a modified root, but it's modified in a way where you have not just the primary effects, but lots of secondary effects. So what we are keen to do is to focus on these later aspects of using patterning, not so much to produce patterns de novo, but rather to limit gene expression. So we have ways of triggering gene expression. It's now a question of how you would have self-limiting patterns. And for this, we've turned to Alan Turing's work in the early 50s, and there are a couple of posters up which are also exploring this idea of using Turing-like patterns to constrain gene expression across tissues, where you use an activator which can promote its own expression, so you have a positive feedback loop, which also triggers formation of an inhibitor, which has a wider ranging effect. So any time that either stochastic noise or some pre-pattern triggers an activation event, then that's self-limiting. And this has now been used to explain a number of different features in biological patterning. And this shows an example of a noise-triggered, a stochastic noise-triggered breaking of symmetry by one of these systems from Hans Meinhardt, who with Albert Gürer essentially rediscovered Turing's work in the early 70s. And so the kind of work that's going on at the moment is, and James Brown is in the audience as well, who's been doing some nice work adapting bacterial quorum sensing systems. And there's a poster from Princeton from Ron Weiss's lab where they've implemented one of these in bacteria as well. And And they're based essentially on the use of quorum sensing systems to allow communication between individual cells, where we have both a positive feedback loop, which is a transcriptional loop, and an exogenous or extrinsic repressing device with a repressor up here, which is communicated between cells. And here we have a hypothetical BioBrick, which is expressed within a hypothetical cell, but that would be implemented within a tissue, a plant tissue. And so the kind of things that James has been working on is to use, in the first case, homoserine lactone-based systems for communication between cells. This shows a plate assay of such a device being implemented in bacteria, and this shows work from the Cambridge iGEM team a couple of years ago using these kind of systems for patterning between cells. And so what we're implementing at the moment, and this is very much work in progress, are these kind of systems now translated into plant systems, we hope. And so, given we're running out of time here, one of the things that we're really trying to focus on in this work are the use of plant systems and trying to take advantage of some of the clear benefits that plants have for translating synthetic biology approaches into a multicellular environment. And there are a number of things that these systems have going for them. First of all, they're easy to manipulate, and any manipulation can be stored easily as seed. There are a number of, as I hope to show, visualization tools which allow us to interrogate and reconstruct cell arrangements directly, and these computer models which allow us to deal with some of the dynamic interactions that are taking place within these systems that we need to model as part of our reprogramming efforts. And of course, plants intrinsically are highly plastic and have distributed organ systems, so that if you reprogram individual groups of cells, as we hope to do, this is a picture of a real plant down here, cross-section of, I think you can see here, lignified tissue around a vascular bundle. And this is you see clear features of plant architecture here, which are essentially boundaries and patterns which are formed during development, which we hope to reproduce. But these are quite, in a way, often quite autonomous, that you can make changes where the entire organ system don't collapse because of mutual interdependence, as you would commonly find in animal systems. You can't take your kidneys away without dire effects, for example. Whereas in plant systems, you can remove whole half of a plant and you can still maintain one part of the system. And of course, with these plants, they're of course make up crop systems. Right, so that's the summary. I just want to thank the people who are involved in the work, and particularly James Brown and up here somewhere, there's Fernand Federici and Jim Agioka and Duncan Rowe, and these three guys here, Tim Roach, Jonathan McKenzie, and Leonard Dupuy, whose program is working on the project. And thank you very much for your attention. So it's probably time for a quick question. Is there one? The question was how does one imagine the non-autonomous quorum sensing system as molecules move between cells? And in the homoserine lactone case, we're relying on movement across membranes between cells. And there's some work using quorum sensing systems from agrobacteria that's been looked at as a signaling system where they had artificial application of those molecules and looked at triggered responses. So we know that there is some evidence that it will work directly. For the longer term work, we're looking at using, and we've already made BioBricks for quorum sensing systems from gram-positive bacteria, which involve small peptides of many different potential channels of communication. So literally hundreds of these things with a high degree of specificity. And they're also parallel, or they're similar to natural systems of signaling that you find existing in plants.